Hello, everybody. This is Miss Peachy again from your WCA biology class. And here we are on Unit 5, Lesson 8, Mitosis and Genetic Information. I did not make a video, by the way, for Unit 5, Lesson 6, Mitosis and Complex Organisms, because it kind of repeated some content that we had previously. So it was really just kind of putting together a couple other lessons in one lesson, and all the information is stuff we already covered in a couple of different video segments. So um, as you go through that lesson, or as you went through that lesson, you probably felt like it sounded very familiar because it should have been. It was all kind of review information. So this one shouldn't be super long. A lot of it is going to be review information as well. Just as a reminder, I do have the kind of outline here for this lesson that is posted in a link beneath the video, which you will find on my website. And just click on that, have it open, kind of take some notes as we're going through today's lesson as well. All right, so again, a lot of kind of review information, but then just a little new information here as well. So it talks a little bit about how complex organisms descend from two parents and their genetic information is inherited from both of them. A fertilized egg will grow and develop, and you'll be able to grow using the process of mitosis. The type of division that is used, mitosis is cell division. We call it mitotic cell division to specifically refer to the fact that this is using the process of mitosis. And you might think that that seems a little redundant because isn't cell division mitosis after all? But we also have meiotic cell division, which uses the process of meiosis, which we have not talked about yet. So it is important to differentiate or distinguish between the two of them. Mitosis, mitotic cell division specifically is for growing, making copies, duplicates, clones, if you will, of your cells. It's not making something brand new. It's not mixing things together. It's just making a copy of what you already have. So that is done in eukaryotic organisms. So eukaryotic organisms is where mitosis is done. And if you remember with mitosis, I'm going to go back to our screen share here. So with mitosis, we've got these phases here on the screen. We've got prophase, and prophase actually, this is divided up into two segments. I have prometaphase on here. I want you to kind of ignore that and realize that this is all part of prophase. I didn't realize that this was a different picture. So we are going to just kind of cross this out here. And we'll just say that this is all part of prophase, kind of early, mid, and late. You can kind of call it a little bit differently. They're calling it like early or before metaphase, which basically is just late prophase. So essentially what happens, remember, is the chromosomes are going to condense and become visible. They have to shorten and thicken so that they can be easily moved around by those spindle fibers. You don't want them to be all long and tangly, so they tangle up with each other and get broken and stuck and things like that. That's bad news. So you want to have them much more condensed. The nuclear envelope is going to break down so that you can kind of have them free from the nucleus. Spindle fibers emerged from the centromeres. So you got this, and then they move towards opposite poles. So the mitotic spindle starts to form. Then the spindle fibers, they have to migrate to opposite poles of the cell. The spindle fibers will emerge from them as well. Chromosomes continue to condense, and we don't have to talk about kinetic cores here. It's not something we talked about, but essentially those spindle fibers will attach, or the microtubules will attach to the chromosomes and start moving them around. They get pulled into the center of the cell during metaphase and kind of line up. The sister chromatids are attached, so then they're pulled into opposite ends of the cell, which happens during anaphase. So now the chromosomes, because the sister chromatids are separated, one half of each sister chromatid migrates to opposite ends of the cell. So you've got a complete set of DNA on both sides. Remember that those Microtubules are also moving around organelles and kind of reorganizing those so everybody gets equal set of organelles. And then eventually the nuclear envelope reforms around our new chromosome sets. The mitotic spindle breaks down, the microtubules break down, and lastly, which we don't have included here, is cytokinesis, where the actual new cellular membrane forms and divides that up into two new cells. So that's just a really, really quick overview of everything we've talked about already, right? So that's mitosis. But that is true for eukaryotic organisms. Prokaryotic organisms do something different. So prokaryotic organisms, these are very simple cells. They don't have nuclei. They don't have their DNA housed inside a nucleus. Also, they don't have other types of organelles as well. Remember, they just literally have like ribosomes, a little circle of DNA, and then they have some free-floating ribosomes in the cytoplasm. So they don't have the other stuff. So they don't have to go through mitosis to be able to divide. Also, they don't have the energy production to be able to put themselves through mitosis. So instead, they use a process called binary fission. So binary fission is a simpler process. It says here a type of asexual reproduction in which a fully grown parent cell separates into two. They're only single cells. So they're literally copying their whole body here, right? Which is just a copying of cell. But they just split themselves into two. And here we have, kind of an outline of binary fission. We aren't going to go through the steps of binary fission. You don't really have to know kind of what happens here. They do have to copy their own DNA as well. So you'd have to go through the process of DNA replication, right? And then each copy is moved to opposite ends of the cell. A barrier forms in the center, and then they cleave and separate. And this can happen much quicker. It says in the lesson that it can happen like every 20 minutes or so. So it's a much quicker process. It doesn't take hours and hours and hours that mitosis takes. There's also fewer regulatory factors. All those checkpoints that we have in the cell cycle really don't happen in binary fission. And so there's much more of a chance of mistakes, and oftentimes those mistakes can be lethal and result in the death of the bacteria. It leads to then more frequent mutations. And so this is also why microorganisms will evolve much quicker than complex organisms, because they first of all, they can reproduce very, very quickly. And secondly, it's much more likely that a mutation is going to happen and not get fixed. So that mutation then gets passed on to the next generation. And if it's not a lethal mutation, it can lead to, just a totally different version of the bacteria in the next generation. Okay, so that is binary fission. So let's go back to the lesson for a moment. So the lesson goes on to say that since single-celled eukaryotes have many controls, including checkpoints, because remember, we still have single-celled eukaryotic organisms as well. This helps to prevent copying errors. A eukaryotic cell's complex structure is in itself an advantage. Cell membranes protect eukaryotes, and their mitochondria provide an energy advantage that allows the cells to grow much larger than their prokaryotic counterparts. Larger cells require more controlled growth in order to prevent errors. Mitosis regulates cell growth, producing more successful organisms. Oops. Okay. So then, as we move on, kind of through some checkpoints and practice stuff and your peer video here, we go to the kind of the second part of the lesson, in which it talks about how during mitosis, when one cell divides to produce two genetically identical cells, when a cell or an organism's genetic material is an exact copy of the parent cell or organism's genetic material, it is the mitotic process that allows single-celled organisms to evolve into multicellular organisms and is what allows cells to specialize. Without mitosis, eukaryotes and multicellular life, the world as we know it would not exist, basically. So it says how Knowing how complex life formed could hold a key to understanding the workings of our own biology. Scientists have believed for over a century that eukaryotes have evolved from simpler prokaryotic ancestors. And this is something, as it goes on to talk about, something called endosymbiont is what they use here, or endosymbiosis. And what they're referring to is something called the endosymbiotic theory or endosymbiosis, where a long time ago, when organisms were just prokaryotic, you have two independent prokaryotic bacteria. One bacteria basically eats the other. Remember, we have the process of um, endocytosis, where the membrane can form around something and then it can make a vesicle and bring it inside of the cell. This can happen with bacterium as well. But in this situation, engulfing this bacteria actually provided a benefit. So engulfing the purple bacteria here provided a benefit to the green bacteria because the purple bacteria was able to do a process called cellular respiration. So one of the things that bacteria lack today are organelles called mitochondria. And a mitochondria is an organelle that does something called cellular respiration, that they can take oxygen and sugar and convert it into ATP and actually be able to provide a more ready source of energy for your body. Bacteria don't have mitochondria, so they can't do this. But certain bacteria can still be able to do respiration, right? On their own. But they don't have like a separate organelle that does this. But if such a bacteria that could do respiration all by itself was brought into another bacteria that couldn't do it, and it provided a benefit to the green bacteria. So now basically the purple bacteria here in this picture could actually make ATP, and that ATP, which stands for, ATP, stands for adenosine triphosphate, and it's an energy carrying molecule. If the purple bacteria could make ATP and share some of that energy with the green bacteria, then you have a relationship that's called symbiosis. And symbiosis is a mutualistic relationship that benefits both parties, right? Think about the fact that right now, inside of your gut, you have a lot of bacteria that are benefiting you. You actually have a whole plethora of bacteria that lives in your large intestines that helps to make you very, very healthy. It helps to synthesize vitamin K, for example. It helps to break down certain foods that you eat to provide nutrients to you. And so it really kind of helps to keep you healthy. You're providing food to the bacteria by eating it. It's living inside your large intestines, and it's providing you with vitamins and things like that. So it's also providing a benefit to you, and that's called mutualism, right? Well, since bacteria have just free-floating DNA, it's very easy for their DNA to kind of mix together and for one strand of DNA to kind of blend with another strand, right? And so what would happen in that situation is that if that would happen, then when the bacteria reproduce through binary fission, the next generation would automatically have all of the information to code to make a new bacteria, a new green bacteria, but also to have it include the little purple bacteria inside of it. So that's kind of the concept behind endosymbiosis, is that you have a situation where two bacteria lived in a symbiotic relationship, and over time their DNA became entwined with each other, if you will. Actually start sharing this DNA. And then as they reproduce through fission, the next generation would produce the green bacteria with the little purple guy inside of it already. It wouldn't have to be eating it to get that symbiotic relationship. And we see this type of incorporation of two DNAs together all the time, by the way. It's called conjugation. It happens where it's kind of how a lot of variety exists within bacteria through this process of conjugation. All right, so that being said, endosymbiotic theory, what the lesson is talking about is like this has been a theory for a very long time, but we've never actually seen this in nature until recently. And in 2020, which is very recently, a group of researchers published a study done on a new archaeon, which is a type of bacteria found off in a deep ocean sediment off the coast of Japan. This archaeon is a single-celled organism, and they named it, I'm going to really try hard here, everyone, Prometheoarchaeum syntropicum, like after Prometheus, the Greek god who created humans and stole fire, right? And so they were actually able to find this particular bacteria. They were actually able to see that this bacteria, Prometheoarchaeum I did better the first time, syntropicum, actually is a bacteria that has this type of symbiotic relationship where it has a symbiont, a bacterial symbiont inside of its body that's able to help it to degrade methane anaerobically. That's what it is. So it's kind of cool because it's the first time they predicted that this should happen. And they actually was the first time they were able to observe it in nature, which I think is also pretty awesome as well. Because that's kind of the mark of a good theory is that you find evidence to support the theory. You actually should make predictions. The theory should make predictions, and then those predictions you can, should prove true over time. So again, that's kind of cool. It's called the endosymbiotic theory, and this is evidence of supporting the endosymbiotic theory. All right, ladies and gents, that's it for this particular lesson overview. If you have any questions, of course, feel free to contact me through Connexus webmail, or you can give me a call here at school. Otherwise, that's the end of this one, so I will see you in the next.